Hello everyone, welcome back. Today we are going to understand gel electrophoresis, one of the most important topic of biotechnology. Also for your NEET examination. Okay. So by the end of the video, you will understand what gel electrophoresis is, its principle, the steps involved, and some important NEET questions. Okay, so let's begin. First of all, what is this gel electrophoresis? Okay, so it is nothing but a technique which is used to separate DNA. Okay, so it is a technique that we use in laboratory. Okay, in laboratory to separate DNA. Clear? So according to its size. Okay, so how does separation occurs? Suppose in your school there are many classes. Suppose class 5, class 6, class 7, class 8, class 9 and class 10. How does the separation occur? By age. Okay, so if any kid is in the age group of suppose 16 years, he will be in class 10. If someone is in 15 years approximately, he will be in class 9. If he is in 14 years of age, he will be class 8, like that. Similarly, in gel, the DNA separation occurs according to its size. Remember it. Now, where are these used? These are used in forensic science, in genetic research, in DNA fingerprinting. So, the uses, write it down. Forensic science, genetic research, and mainly DNA fingerprinting. Okay, so now let's see the principle. What is the main principle? How does it get separated? How does the DNA get separated? By their sizes. How? So what is the principle working? There have to be a principle now. So we all know that DNA contains what? A phosphate group. Okay, now you will be saying that DNA also contains a nitrogenous base, a pentose sugar, right? But in case of gel electrophoresis, we are focused only on the phosphate groups. Okay, so this phosphate group gives a negative charge. Remember it, it is very much important. This phosphate group gives a negative charge to the DNA. So negative charge is given biphosphate group. Remember it. Okay, I'm highlighting it. Clear? Now see. So what happens when electric current is passed in these negative charges? When electric current is passed in these negative charges of DNA, the DNA runs. Okay, and where does it run? Now I will show you an image. This is nothing but the image of a gel. Okay, this is a gel. See, you can very easily understand it is a jelly like structure. See, the whole thing is a jelly like structure, and these are bands of DNA. These are bands of DNA. These are bands of DNA. Okay, now when electric is passed through a gel, DNA moves towards the positive electrode, also called the anode. Now what happens? As I mentioned earlier, DNA was negatively charged. Okay, and it is very obvious that DNA, suppose this is the DNA will go from negative to positive charge. When will it go from negative to positive charge? When you give electricity to it. Okay, that is from where? From a positive electrode, to a positive electrode, that is the anode, from a negative electrode, that is the cathode. Okay, so the main principle is the DNA, when we apply a charge or electricity, okay, the DNA will move from negative charge to positive charge, that is from cathode to anode. Okay, so it is very easy that if the size of the DNA small, it will run fast. Now why? Suppose a race is going on and there is a bulky guy. Okay, here is a fat guy and there is a very thin guy. Who will run fast? Obviously the thin guy will be the winner, and the fat guy will lose. Why? The reason is its weight. Similarly, if the DNA is small, the DNA will run fast in the gel. Okay, as compared to a heavy DNA molecule. Now see, this is the gel. This blue one is the agarose gel. Okay, and these are wells. What are these? These are wells. Inside these wells, there are many wells inside the agarose gel. Okay, so inside these gels, we insert the DNA. There can be many types of DNA. You know, all the genes are nothing but DNAs. So we inject or insert the DNA. Now the DNA will run from negative end to positive end. Now it is very clear that if the molecular weight is very high, see, Suppose it is 10,000. See, this is the 10,000 one. The 10,000 band is coming over here. Why? It is unable to move forward. Okay, it has just moved this much. Now, if it is 500 only, so 10,000 and 500, there is a huge difference. The band will go over here. Why? The molecules are less and small in size. It will run much faster. Okay. So this is the logic, or this is the principle by which the gel electrophoresis occurs. Okay. Now see what things we will be needing. We will be needing a agarose gel. Okay. Now what is this agarose gel? Agarose gel is nothing but a jelly like substance which is made from agar. Okay. Now agar we get from Gelidium. Gelidium is a type of algae. Okay. If you don't know, you must know this. Now inside this agar or the gel is kept inside a buffer. Okay, it is kept inside a buffer, so it can conduct electricity easily. Clear? Thirdly, the main important thing, the main important thing is that we will be needing DNA samples. Obviously, to run the gel electrophoresis, we will be needing the DNA samples. Okay. And then comes what? The current electricity. Yes. Without current, is it possible? How will it move from negative to positive charge? Without current, as because current will help this DNA to go to the negatively charged DNA to go to the positive charges. Okay. So this will allow, the fourth one is the current or electricity. Okay. Which allows the movement. Okay. So after we have inserted the DNA inside this gel, inside the gel, we will give electricity. We will run the gel. This process is known as the running of gel. After the running is over, we will again be keeping this whole gel. This is, see, this blue hole portion, this whole portion is the agarose gel. Okay, it is a jelly like thing, jelly like structure. Okay, so with this gel we will be keeping this whole gel in a tray. Okay, suppose this is the agarose gel, and the tray will be filled by ETBr or ethidium bromide. It is known as ethidium bromide. Please remember, ethidium bromide, which will stain the DNA. Okay, you don't think that these bands will be visible in your eyes. No, these bands will never be visible. Okay, to make it visible, we will have to keep these DNA bands which are inside the gel into this ETBr or ethidium bromide solution, which will stain the DNA. Okay, please remember, this ethidium bromide will stain the DNA. Okay, after staining, what will happen? We will be transferring the gel or the agarose gel. This was the agarose gel. Okay, so there were DNAs over there, there were DNA bands, there were DNA bands. Okay, so this will be transferring the gel into UV trans illuminator. Okay, so we will be just transferring it to the UV lights so that this flourishes, and you will be able to see this is the UV light. Okay, and you will be able to see these bands. See these bands, it looks so good. See, these are the bands, these are the bands, these are the bands. Okay, these are the bands, these are the bands, like this, like this. Okay, these are the bands. See, these are the bands. So that is the thing. Okay, so firstly we prepare the gel in the first place. Okay, then we load the DNA. See, firstly obviously we will have to prepare the agarose gel, then we load the DNA, then we give electric current. Okay, then after giving the electric current, the DNAs will separate, very obvious it is. Then the last process is known as visualization. That means to visualize or to see the DNA. Clear? So the first question, these all are NEET level questions. In which direction, I am writing it for you, in which direction does DNA move during gel? Very easy question. Okay, you all just pause it for a second, think, and then verify with the answer. Okay, so the DNA moves from cathode, that is negative, to anode, that is positive. Remember, from negative electron to positive electron, cathode and anode. Okay. Okay. Another question. Okay. The next question comes. Just discussing 2-3 questions. Okay. Very easy topic. If you just see the video, it will be very helpful for you, and just share with your friends. On what basis DNA fragments are separated? Okay, so the basis is very easy. They are separated by based on size or molecular weight. Okay, so based on their size or molecular weight, they are separated. So I think you have understood about today's topic. It was not that easy topic. It is a bit of critical, I would say. So thank you for watching. Share my videos.